In this video, we're looking at protein synthesis. We're looking at how DNA gets transcribed and then translated into an amino acid chain or a polypeptide chain. So DNA, as we described in DNA replication, can replicate, completely copy its entire library in the nucleus to make itself into two identical cells. Now, in this case with protein synthesis, we're looking at two phases. The first phase is called transcription. This is where DNA, because DNA is such a large molecule and it never leaves the nucleus, that important information needs to get transcribed so that it can leave the nucleus and go into the cytoplasm. Now, unlike DNA replication, we are not going to make a copy of the entire library of the entire genome. Here, we're just looking at a gene, a specific gene in the entire genome. So if you recall, we said that we can make about 6,700 or so books of information from the genome. We might only need one book or one page to make one amino acid or one particular protein. Just like a liver cell, even though all liver cells have all of the genome in them, they're only going to copy the proteins, the genes that they need for their particular functions. So we're only looking at a small portion of the whole genome that we need. So that's going to get transcribed. Okay, we go from a four-letter alphabet of DNA to still a four-letter alphabet code in mRNA. The only difference now is that instead of having thymine, we're going to replace that with uracil. And now mRNA can leave the nucleus and it will go into the cytoplasm where then it will get translated into an amino acid chain or proteins. This is where the translation, this is where the language completely changes. We go from mRNA that has adenine, thymine, cytosine, guanine, adenine, uracil, not thymine, adenine, uracil, cytosine, guanine, a four-letter code to now a 20-letter code, if you will, of the 20 different amino acids that make up the proteins in the human body. So this is where the translation, this is where the language completely changes. So let's go ahead and get started. We're back at the nucleus and we're looking at a DNA molecule. We need this particular gene in the genome. We need this particular code for this from this DNA molecule. It's kind of looking at a recipe book. I only need one recipe, so I'm only going to go to one or two pages. So what's going to happen first is that we need to unwind this molecule, and we're going to have an RNA polymerase that's going to show up, and it's going to start to transcribe each one of these nucleic bases, and it's going to put down the complementary nucleic base on only one of the strands. It's going to completely ignore the other one and transcribe one end of that DNA molecule. And notice again, with adenine in the mRNA molecule, you no longer have thymine, but you have uracil replacing thymine. So now I have that one single strand of mRNA, messenger RNA, that's a single strand and has uracil in it. It's going to now leave the nucleus, and as I mentioned before, it will go into the cytoplasm, and it's going to meet up with a couple of other molecules called rRNA and tRNA. So all of these molecules are made up of RNA, okay? No DNA here. All the DNA is in the nucleus. Every single one of these molecules is made up of a single strand of RNA, and they're going to kind of spaghetti their way into forming these particular molecules like rRNA, ribosomal RNA. These are your ribosomes. rRNA is a multiple complex system that has different RNA strands that come together and make a functional RNA. Now this rRNA is the part that actually brings both the mRNA and the tRNA together. So if you consider the tRNA, for example, the tRNA has your amino acid at the top and then it has the anticodon at the bottom. That anticodon coincides with the codon in the mRNA. So let's see how this goes together. So as I mentioned before, the ribosomal RNA is going to put the two together. You have three different sites in the rRNA. You have the APE, the reverse ape there. You have the A for arrival, P for where the peptide bond is made, or you can think processing, or maybe drops off the passenger, and then the E for the exit site where it takes off. So think of an airplane landing on the A site, and then the P part is where it drops off its passenger, and then the E is where it exits, it takes off. So landing A, processing P, take off E, the exit site. So the A acceptance site, you'll notice that there are 3 nucleic bases that are there because it reads it in groups of 3. So the adenine uracil guanine, the anticodon of this codon, is at the tRNA, and you'll see that if you go back to the tRNA, you'll notice the uracil, adenine, and cytosine. These correspond with the 3 in the mRNA. And again, notice that it has a particular amino acid. As the mRNA is going to go through the ribosome, it's going to read it, and it's going to move the tRNAs into each of their positions. So as it goes from A to P, its new position, you'll notice that A is exposed. And now a new 3 codon is exposed, and that codon, that cytosine, guanine, and cytosine, correspond with a new tRNA that has a new amino acid. Every single one of your 20 amino acids has its own anticodon, its own code that codes for that specific amino acid. And remember that the order of these amino acids is crucial for having a functional protein. Just like in the English language, we have letters in our alphabet. And if we don't put those letters in the correct order, we don't convey information. We don't convey a message. Likewise, when we're reading DNA code that gets transcribed into mRNA, we are actually looking at a language. We're looking at a code that conveys information. And that information is the order, the specific order of all these amino acids that make a specific protein. So the tRNA, it's going to land in the acceptance site, and then you'll notice at the top that it's going to make a peptide bond. So the nitrogen groups in each one of these amino acids are going to extend, shake hands, and make a bond. And notice what happens as the mRNA continues to move to the left. Each one of these tRNAs are going to move over to they're going to move over to their next site. And notice what happens to the amino acids. They bond together. And look at that first tRNA. It has lost its amino acid. It will now be ejected, and it will go back into the cytoplasm to pick up a new methionine, a new amino acid. And again, I have now exposed the A site again with a new code for a new amino acid. And then they're going to make a new peptide bond, and that amino acid chain at the top is going to get longer and longer. It's going to elongate. Okay, this is called elongation. Now notice on the far left, you have that tRNA, it got ejected. It's off it goes. And then the new one on the exit site will then get ejected as the mRNA code continues to go and move along. So here's a new code, the new anticodon with the tRNA, dropping off its enzyme. And this process continues for as long as it needs to until the amino acid is satisfied, until that amino acid will become the new protein. So again, here we go. So the mRNA is going to go through the rRNA, and the tRNAs are going to drop off their enzymes. And notice what happens to the amino acid chain as the mRNA continues to go. It gets longer, it elongates. Let me back up real quick so you can see that again. So mRNA is going to go through the ribosome, and as it goes, the enzymes are going to make amino acid chains, and they're going to get longer, they're going to elongate. And that amino acid chain will get longer and longer, and it will then become a protein later on. So if I center it here, these amino acid chains, recall that amino acids can be as short as they need to be, anywhere from 3 amino acids all the way to 45,000 amino acid long. So depending on the protein, depending on the size of the protein, okay, it's going to determine how long that amino acid is. And recall that the order of the amino acids is crucial to making the functional protein. These amino acids will start to shake hands. They will make bonds, taking a very specific configuration, and then they will make the protein. And then that protein, like I said, will then go to whatever portion of the body it needs to go. It might become part of the phospholipid bilayer. It might become part of your cornea. It might become part of your muscle. Your muscle fibers make up some of the largest proteins in the body. In fact, the longest protein in the body that we know of is in the muscle groups.